Okay, everybody, I think we'll make a start if we could. My name's Scott Johnson. I'm co-convener of this Thematic with Will Hentley here. So we're going to join the chairing of our thematic session on invertebrates and global climate change. And we use the prefix, which little things will run the world, with reference really to that phrase by E.O. Wilson, the conservation biologist, about invertebrates being the little things that run the world, and essentially was talking about all of the ecosystem services that they provide. But of course, invertebrates cause a great deal of damage to crops and to human health. So we thought this thematic was important really from the point of view of understanding how climate change might affect some of these little beasties that really do influence our planet. So we've got an exciting lineup of speakers, but I'd like to kick off with our keynote speaker, who's come all the way from Canada, from the University of Gelton. That's Jonathan Newman, and he's going to speak to us on invertebrates and global climate change, progress and perspectives. Thank you, Jonathan.
Right, well, I want to say thank you, Scott, and thank you, Will, for organizing this session, and thanks to the Hawkesbury Institute for sponsoring it. I'm very pleased to be able to contribute to it. So I began working on climate change in the early 1990s. I wasn't particularly interested in it, actually. Some colleagues of mine asked me to collaborate on a grant proposal. At that time, climate change, if you'll pardon the pun, was not quite the hot topic that it is today. It was, it was somewhat of a fringe topic in ecology, but I liked my friends and I wanted to participate, so I started doing some reading, and I read this book edited by Kariva et al. And in that book, they laid down a challenge, and if I can paraphrase, the challenge says, if we as ecologists cannot predict the patterns of diversity change associated with the global onslaughts, such as climate change, one has to wonder just what ecology can predict. And I thought that was fair enough. I think of ecology as a quantitative predictive science, and I think it is something we ought to be able to do. And so that has largely motivated me for the last 20-some years. So what I want to do with the rest of this talk is I'm going to elaborate a little bit on what I think, how I interpret that challenge, talk a little bit about what I think the answers to that challenge might look like. Then I'm going to step back a bit and ask how we as a field of ecologists have addressed that question. Have a little bit of perspective on how I think we've done at answering it, and I'll have a few thoughts of how to go forward. So this is a caricature of what I think the challenge is, and I want to hasten to add that you are under no obligation to agree with me that this is what the challenge is. We may rightly interpret the challenge differently, but this is how I do it, and it's going to motivate a lot of what I say, so I think it's important to spend a few moments on it. So I think of the challenges like this. You've got some insect response variable in the blue that changes over time, let's say over the course of a year. The time scale's not very important. And it responds mechanistically, either directly or indirectly, to some parameters, P1 and P2, that are normally found in this parameter space here. And sometime in the future, they're going to be found in the parameter space here. And our goal is to try to say, well, what will happen to this response variable as a result of this? With climate change, there are a lot of contenders for what P1 and P2 might be. Could be carbon dioxide, could be temperature, could be relative humidity, could be precipitation, could be UV, could be ozone, could be climate variability, and so on and so forth. So in fact, this isn't really a two-dimensional problem, it's an n-dimensional problem. Also point out that the difference between P1 and P2 in the future and in the present might itself be different at different times during the year. It almost certainly will be different in different places on the Earth, adding to the challenge. So if I just develop this a little more, this is the climate projections for the city where I'm from, Guelph in Ontario, Canada. You see here in the blue, this is the normal range of climate contemporarily, the baseline, and in the sort of peachy color here is the projections. You see that, for example, in the winter, there's a much bigger difference between the temperature minimum than there is, let's say, here in the early summer, and a large difference in the temperature maximum in the summer, but not so much, say, in the winter, and so forth. On the right, you see precipitation, the normal distribution of precipitation, and here's what the projections look like. So in Guelph, generally speaking, it's a bit wetter in the winter and spring, and not really much of a difference at all during the growing season. But none of that would be true if we looked at other places on the Earth. So if I wanted to predict something about what climate change was going to do to this insect response variable, I would want to look at this sort of parameter space. So these are the differences in the daily temperature, mins and or maxes. Here are the differences in precipitation, and climate change is going to be somewhere in here, and I would want to sample that area for this response variable, and I would want to do that for a whole range of carbon dioxide concentrations, because there are a whole range that it might be in, so anything from about plus 50 contemporarily to about plus 900 contemporarily. So that's really a three-dimensional space here that I want to sample. And if I were to test some combination of these things that's not in this parameter space, I might well learn something quite interesting about the biology of the system, but I'm not sure that I would be saying anything particularly relevant to what climate change is likely to do here in this system. So how do we answer Kariva et al.'s challenge? Well, on the one hand, you could have general answers, right? Your general answer, or general questions that produce general answers. Your general question could be things like, will insects be more abundant or less abundant? Will insect ranges expand or contract? Will diversity increase or decrease? And so on and so forth. And sure, there's going to be deviations from those answers that we give. It's not going to work for every single organism in every single place, but there's probably some generality there. And we could fill in those details if we had to. But we would be giving broadly, what I would describe as broadly descriptive answers to that challenge. And I would argue that those things are marginally predictive. So an example of that, meta-analyses, we now have a large number of studies, and we can start aggregating them into meta-analyses. And this is just one that my students did. There are many others out there. So if we look at the proportional change in abundance under elevated CO2 and just look at the green dot here, you see that there is, so the general answer is there is no effect on abundance under CO2 on average. That's an answer of sorts. Some of you might protest. You might say, yeah, because, well, many of you probably work on rising temperature, for example, because that's a more common topic in climate change. And you might say, yeah, but what about temperature? And I might say, well, yeah, well, maybe, what if this was a meta-analysis about temperature? I would say, well, what about CO2? So spoiler alert, there's almost no work on interactions among climate change factors. Very, very little of it. Most of it, the vast majority of it is just single factor. I think it's also worth pausing to always remember when you're looking at a meta-analysis that meta-analyses suffer from at least 3 problems. They suffer from the file drawer problem, which is just all those papers that the results were not significant and therefore never got published. There are some statistical ways to try to control that. The other 2 problems are a bit more serious. One of them is called the apples and oranges problem. It's on display for you here, is that we are doing a meta-analysis over many different kinds of insects here. Is there any reason to believe that all insects should respond similarly? Does that average even make any sense to talk about? And the last problem is what's sometimes called the flat earth problem, which is you're encouraged to forget about any statistically significant results that happen to go in the opposite direction of the meta-analysis here. You should just pay attention to the conclusion from the meta-analysis. So of course, when we dig deeper into this problem, we see that yes, in fact, when you split it out by orders or by feeding guilds, there are some statistically significant responses to elevated CO2. Some of them are negative, like the Lepidoptera. Some of them are positive, like the Homoptera, and so on. But don't be fooled by that. The same 2 problems exist here. This is still an apples and oranges problem, and it's still a flat earth problem. There are still significant results that go in the opposite direction, and I'm still averaging over many different kinds of insects all at once. But nevertheless, that's what this is an example of how we can give general answers to general questions about whether they'll increase or decrease, and lots of other response variables too, not just obviously not just abundance. On the other hand, we have specific questions that would require some kind of specific answers. So, for example, will mountain pine beetle be more abundant or less abundant? Will mountain pine beetle still be found east of the Rockies in Canada or not? And so on. Those kinds of answers are narrowly quantitative answers, and generally speaking, we're interested not just in predicting some response, but we're also interested in characterizing the error around that or the uncertainty around that. I'll just illustrate that briefly with this problem. This is spotted wing Drosophila. It's a recent invader to North America and to Europe. I didn't work on this at all, but the Ministry of Agriculture came to me and asked if we could do some work on it because they were worried about it as an invasive species. Because it wasn't well known, and there wasn't much research on it, we started off by doing a life table analysis. And I have to say, as an aside, I have never felt more like a real ecologist than doing Drosophila work and doing life tables. These are things you read about in general ecology textbooks, but you never actually get to do. So we did a life table. We then looked at how fecundity changes with female age. We looked at how fecundity changes with temperature. Don't worry, you don't have to get any of the quantitative results out of this. We looked at how development rates changed with temperature. And then we were able to piece together a picture of the life cycle. We're able to then turn that into a conceptual model stage-based population dynamics model. We then translated that into a series of coupled differential equations. And I'm not going to spend the next 20 minutes talking about those. You'll just take my word for it. We then ran the model for these locations in North America because these are the most important production sites for strawberry and raspberry commercially in North America. They're counties and townships. And strawberries and raspberries are among spotted wing's favorite hosts. If you just concentrate, for example, for a minute on the green bars here, these are the normalized yearly number of flies, and notice it's a log scale. So here's Ontario, where the ministry that came to us was interested in, and you can see that it's about 2.5 orders of magnitude less than where we would expect the most productive sites in North America to be. If you look at the orange bars for a minute, that's where we added a simplified fruit model to sort of account for fruit abundance and availability. And when you do that, then it goes from about 2.5 orders of magnitude to about 1 order of magnitude different from the most productive spot, which is now in New Jersey. We then expanded this to look at what do we expect under climate change, and we expanded it to look across the entire continent. Obviously, I'm not showing you the whole continent here, but what you're seeing are consensus maps that are developed from a bunch of different climate models, and I'm showing you for 3 time periods for 4 different climate change scenarios. And just as a qualitative result, you can see that as you move down and to the right, the graphs are getting less intensely orange, which means they're not as good a climate for population growth of this fly. If we look a little more quantitatively, these are the same counties and townships I showed you before. This red line is the historical climate. And you see there's a few places like in North Carolina, where under climate change, we expect considerably less population growth. And then there's places like in Ontario, where there's considerably more population growth. So these are the kinds of answers we were able to provide to the ministry, but they come with some serious caveats. So first of all, we don't know what natural predation rates are in the field for spotted-winged Drosophila. It's not an easy parameter to get to in the field, which is arguably the only place where that parameter can reasonably be measured. But we were able to show with the model that it matters quite a lot. So this is just sort of increasing predation rates, and this is the change. Remember, it's a log scale, so a little bit of predation goes a long way here in controlling this pest. We don't have any adaptive evolution in here. We certainly know that insects are capable of adapting quite rapidly if there's a strong enough selection pressure. Think of insecticide resistance as a good example of that. And we don't have any effects of elevated CO2 on larval growth. Probably elevated CO2 does not affect larval growth directly, but it certainly affects fruit quality, and that affects the larval growth of the fly. So, we can make these kinds of statements for the ministry. We could say, well, spotted wing is probably not as big a problem in Ontario as it could be in other places in North America currently, but it's probably going to be a much more significant problem under climate change, with those caveats still in mind. Let me take those are illustrations of kind of the general problem and general answers, and specific problems and specific answers. Now I just want to back off and say, what did we do as a field? How did we as a field respond to the challenge that Kariva et al. Laid out? So I did this literature search, rather overly ambitious, I'm afraid, just climate change and global warming and insects. In Web of Science, came up with a bit more than 3,300 papers. I then started eliminating all those papers that really weren't about insects. They were about plants, or they were based on tree rings or fossils, or they were reviews, meta-analyses, opinion pieces, and so on. Or, and this is a big one, where climate change or global warming was a sort of throwaway afterthought to the work that they did. It's sort of like, well, we did this stuff, and you know what? It might tell us something about climate, motivated by or driven by that. Doing that, I have eliminated almost 1,900 papers so far, and I have kept 275, and I still have about 1,000 to go. So, take these conclusions with a grain of salt, but I think it's a reasonably good sample of what's out there. I classified the work that was done by the kind of question that the people were trying to answer. So were they trying to answer population questions? Were they trying to answer community questions? Physiology, phenology, behavior, or evolution? And those are some examples of the kinds of response variables people were looking at. And when you do that, you see that the literature is very heavily dominated by population studies. Mostly what people are looking at are population responses of some kind. And what they're really not looking at is evolution, as a relative number, certainly. If I look at the kinds of studies that are done, about half of them are field-based studies. About a quarter of them are chamber or lab studies, and about a quarter of them are theoretical papers. A small amount are greenhouse-based studies. In field studies, if we just look at the field work, the vast majority of studies are using natural variation. So these are things like time series, latitudinal gradients, elevational gradients, other forms of spatial variability. As a sort of analog of climate change. There are about a quarter or so of the studies use actual manipulations. Those manipulations tend very heavily towards temperature. They use some sort of form of passive warming usually, or towards precipitation manipulations that, generally speaking, increasing drought. And then a small number of them are just looking at climate oscillations. Chamber and lab studies are very heavily dominated by temperature, just looking at temperature by itself. I've got that asterisk there to just remind me to say to you that those fly studies I showed you are exactly the kinds you see, except our studies didn't show up in my search because we didn't mention climate change at all in them. But a lot of those studies that say they're motivated by climate change are doing exactly what we did there. Then a much smaller proportion are doing CO2, and then you've got a very tiny grouping that are doing some kinds of interactions between CO2 and drought or temperature. The theory papers are very, very heavily dominated by species distribution models. These are species distribution models in the dark blue. In red, you've got physiologically based mechanistic models, and in the light blue, you've got other, where other is largely but not exclusively statistical modeling, a lot of regression kind of stuff. And the asterisks are there to remind me that, for example, the population model I showed you of spotted wing would fall into either of those because we were looking at both abundance and spatial distributions. Okay, so I know that was a really fast romp through that stuff, but a couple of things that have become clear to me in looking at the literature are that fieldwork is dominated by natural variation, and I think we always have to remind ourselves or ask ourselves the question, is that really a good mimic of what we expect under climate change? Experiments they never test specific climate change projections, and I would argue, nor can they. It's just logistically impossible to do that kind of multidimensional problem. Theory can do that, but it's dominated mostly by species distribution models. There's almost no behavior, and I didn't come across yet any community level theory. So if there are any theoreticians out there looking for problems, that would be fertile ground. And as I said before, there's almost no work on interactions. Conceptually, I think we need to fill some gaps. We need to stop looking only at single factors and equating this with climate change. That may have been fine when we started the work. Now I think we've got to move on from that. Constant manipulations, like we're going to look at temperature or temperature plus 2 degrees, that's not actually a mimic for what climate change is expecting. The experimental conditions can't approximate climate change projections, but models can do it, but most of them don't. It's very, very few that actually try to take into account the whole vast panoply of climate change. I would say we're quite good at collecting the parts that you need to put all this stuff together, but we're not very good at putting it all together. You rarely see that followed through. And, by the way, if that sounds critical, I'm including myself in that. I mean, I'm not any better than anybody else at this. So how have we done? Well, I would argue we've made some progress, but it's been kind of spotty. Our work, when you survey it from afar, when you take a step back from it, seems kind of haphazard, actually. We've shown some evidence. We've shown some evidence of some things, and we've not really put it all together very coherently. And I haven't shown you any evidence of this. You're just going to take this claim at face value, and we could argue about it if you want. But when I survey the literature and I look at all the claims that are made about climate change impacts, I think actually very little of that literature is really speaking directly to the kind of answer that I sketched for you before about what I think Kareev et al. Have challenged us to do. So lastly, just conclude with a couple of thoughts. I think a way forward would be for us to behave more like climate scientists and less like ecologists. So what do I mean by that? When climate change first kind of reared its head, climate scientists responded as a community. They got together, they decided how they were going to divide up the work, they decided how they were going to report the work, how they were going to archive the work, to make it all parts of a picture that all fit together, that give you a coherent answer. Ecologists responded much the way Bob May has described the way ecologists always respond to crises. We circled the wagons and then we shot inwards is what we did. We don't collect our data in a consistent way. We don't archive it in a consistent way. We don't document it in a consistent way. We haven't had any serious discussion as a community about how we should go about solving, providing answers to Kareev et al.'s challenge. Nevertheless, we've kind of stumbled along to some answers. I don't want to leave anybody with the impression that we don't know anything about climate change here. But I do want to point out that even if we did what climate scientists have done, our task, I think, is actually considerably harder. I doubt we're ever going to be able to provide quite the same kinds of answers that climate scientists have been able to give us about climate, partly because biological systems are just inherently more complicated and more noisy. We've done pretty well at giving general answers of the sort of meta-analysis and others that I've shown you there. And I want to just pause and pose the question of whether that's enough. I think when the Ministry of Agriculture came to me and asked me about spotted-winged Drosophila, I think if I'd have said, don't worry, on average, insects are not going to increase under elevated CO2, that's not really a sufficient answer. And I think a lot of policy work really depends on that kind of more specific study of the impacts of climate change. And so I've finished with enough time, I believe, to have some questions. I just want to thank my funders and the people who really did the work. So thank you very much.
Thanks, Jonathan, for a really excellent keynote presentation there. As Jonathan says, we've got time for some questions. Anybody has any? Maybe I'll start the ball rolling.
Sure.
So you mentioned a couple of times there the importance of testing multiple factors. And a kind of, I guess, is a logistic problem to doing that, because as soon as you add another layer of treatment, so let's say you had temperature by CO2 for replicates, you need 16 chambers instantly. And few of us have got sort of access to resources like that.
Yeah.
So sometimes I guess when you submit papers, there's an element of pseudo-replication and things like that. Where do you think the balance should lie?
Good question. I didn't point it out, but in those chamber studies in the lab, that's even a bigger problem. I mean, and again, I'm just as guilty of this. You have access to two climate chambers, two environmental chambers. One is your elevated temperature and one is not, and there's ways you try to get around that by swapping the replicates back and forth between the chambers on a regular basis, but never really gets you out of that problem. I mean, yeah, in short, logistically, it's almost an impossible problem to solve without the kind of money that, I sound like an envious scientist here, but the kind of money that was thrown at climate change, the climate scientists got a lot of money to study this, and without that kind of money, I mean, we have poured some serious amounts of money into things like some of those face studies. It is just expensive stuff, and there's only been a few of them. They haven't gone very long term. And they're not, even then, they're not terribly well replicated. I mean, I think where we overcome that is through the mountain of individual work, right? So yeah, we're all slightly guilty of pseudo-replication. I can feel like I'm in an Alcoholics Anonymous meeting. Hi, my name is Jonathan, and I'm a pseudo-replicator. We're all slightly guilty of it, but I think that in the mass, as we all do this and we bring it together in things like meta-analyses or as parameter estimates for models, we get around that problem. It never really goes away.
Absolutely. Any other questions?
There's one over here.
Oh, sure.
It's sort of more of a comment. First, I absolutely agree with you on the fundamental differences between the climate science community and the ecology communities. It would be wonderful if we could have the kind of coherence that the climate scientists have. But, and I agree with your reasons, except I'd add one, is they're a really small community.
Yeah.
I think that's an enormous help. They literally can all get together in a room this size.
That's right. And they're also, it's such a small community, they're all kind of trained, they all come out of the same labs, right? You're right, it's a very small community, and so it makes it more feasible.
And also I agree with you that also fundamentally biological systems are enormously more complex. And I'm not saying we shouldn't keep trying, all of us, but we are never going to get to the level of efficiency and accuracy that the climate scientists are.
Absolutely, but I think you probably would agree there's still room to improve on our side. Yeah. And I would also just add, when Bob makes his comment about circling the wagons and shooting inwards, he's usually comparing it to physicists, where they are a large community, and yet they manage to get things like the Large Hadron Collider done. Bob's argument is that they do that because they are a friendly, supportive group who says, yeah, jolly good, that sounds great. Let's everybody support that idea, and it happens, whereas we are a highly competitive group, and we don't get that kind of support financially.
Okay, well, we're just about time there. Maybe things will get better under Trump, I don't know. Thank you very much, Jonathan, for that. Sorry, Julia, I didn't get to you there, but maybe you could ask Jonathan at a break. Okay, so we're moving on now. I'll just give people a chance to come into the room. So it's an enormous pleasure to welcome Professor Jane Hill from the University of York, who's going to speak to us about climate change and range shifts.
Thanks very much, Scott, and thanks very much, Will, for organising this meeting, and thanks for inviting me. So I'm Jane Hill from the University of York, and I'm going to be talking about some of the work that we've been doing in the lab, where most of the people working in my lab in York have been focusing for a long time on Lepidoptera. So I'm going to be talking mostly about Lepidoptera and climate change. And I suppose when it comes back round to the title of this symposium, I think I'd find it quite difficult to argue that Lepidoptera are important in running the world. And I guess maybe important, some of them, certainly for the moths, in terms of being bird food and in being important pollinators. So that's quite an interesting thing to talk about, and interestingly, very few people ever question why it's worth looking at butterflies. They just take them as intrinsically interesting, which which is great. But one of the things I do think is important is actually Lepidoptera have been ruling the world in terms of thinking about climate change research and thinking about the ecological impacts of climate change. And there's a number of reasons for that, primarily because there's such fantastic records. So one of the reasons why there is so much work being done on Lepidoptera is we've just got fantastic data going back in the UK over the last 40 years. So those are the data that I'm going to be talking about. And when we're talking about butterflies and moths, and essentially the data that I'm going to be presenting are either distribution data, which are sort of citizen science data, information about the occurrence of individual moths and butterfly species at particular locations. So when I'm talking about distribution, that's what those data are. But the other thing is that we've also got lots of abundance data as well. So these are fixed effort data from light traps and from transects. So the great thing about Lepidoptera is that you've got both sets of data. You can look at distribution, you can look at abundance, and by the time you combine butterflies and moths together in the UK, you've got a reasonable number of species to work on. So that's what I'm going to be talking about. So we know that when we're thinking about how species are going to respond to climate change, we put them into these three broad categories. You either shift your range and track climate, you either stay put and adapt to changes in situ, or you go extinct. And I'm going to be focusing on the first of those today, thinking about how species shift their ranges and track climate change. So I'm going to only be talking about species that are expanding and shifting northwards and essentially, if you like, benefiting from climate change rather than thinking about things that are going extinct. So we are at the moment in the lab thinking about adaptation to climate change. So we are thinking about genetic adaptations and phenotypic plasticity. That's a project that's just started, and I'm not going to be talking about that in much detail today, but do come and talk to me if you're interested in it. And I'm not going to be talking about another project we've got going where we're running a survey of northern butterflies looking at range extinctions. And this is a resurvey of work done we did with Aldina Franco 10 years ago, and that's an ongoing study as well. And again, I'm not going to be talking about that, but if you want to hear more about that, come and talk to me, or you should go and talk to Andy Suggett, who's also at the conference as well, who can tell you a lot more about it. So we're going to be talking about range expansion. So this is an example of a species that's expanding its range and gives you a sort of an idea of the sorts of information that we have in the UK to look at how species are changing their range. So this is one of the most impressive butterflies, if you like, that's shifted its range in the UK. So this is the comma butterfly. So if you live in the UK, you're probably very familiar with this species because it turns up in your back garden, or it turns up on your allotment, or you just see it around in urban environments a lot. So this is shifting its range pretty rapidly. Each of those green dots on the map there is a record within a 10-kilometre grid square. So this is the sort of types of data that we deal with at this sort of resolution, sometimes at much finer resolution. Now that everybody's got a GPS on their smartphone, the data are coming in at much, much finer resolution that we analyse. But that's the sort of example of a species that's shifting its range and moving northwards in the UK over the last 50 years or so. And the great thing about the comma butterfly as well is that it very handily shows us from its morphological variation whether or not it's actually breeding locally at the site or not. So you can separate out things that are just very mobile individuals that are dispersing in from those that are actually breeding locally. So that's an example of a species that's doing particularly well and shifting its range and expanding northwards in the UK at the moment. So that example gives you an idea of the sorts of types of measurements that we take. So using those data again, so each of those dots at a 10-kilometre grid resolution, you can start to draw out specific measurements of understanding responses of species. So in this particular case, it's the distribution records, and we're interested in change in the extent of the area. So the sorts of data that present are comparing the black and the grey squares. So what was its area of extent in an earlier time period compared with a later time period? In this case, for a species expanding its range. And also, we estimate another variable, which is where the location of the northernmost part of the leading edge of the range margin is. So that's shown here for the earlier time period, which is the latitude of the most northerly 10 10-kilometre grid squares, and then where it's shifted to at a future time period. So you'll see data where I talk about the distribution change, so that's the change in the area from black to grey, and also margin shift in this case, which would be from the black line up to the grey line. And of course, we also look at abundant changes as well. So this is a graph that I'm sure you're very familiar with. We keep coming back to it a lot because it shows a lot of features of our studies very well. So this is no longer just looking at Lepidoptera, but looking at a vast number of different taxonomic groups that we have data with that we can analyse. So this is work done by Rachel Hickling back in 2006. Don't worry very much about the different colours of the bars. Essentially, that's to take into account differences in recorder effort. But what I hope you can see is that most of these taxonomic groups are shifting northwards, not all of them. One taxonomic group here that isn't. And also another bit of information, of course, is the degree to which things are lagging behind the rate of climate change. So this red line is just put on approximately here at 120 kilometres north. So anything that is above zero shows its range margin has shifted northwards in the UK over the last 40 years or so. And this is what we estimate they should have done if they were keeping perfect track with the warming temperature. So lots of things are heading northwards. Not everything is keeping track, and a few things are declining. But what I hope you will get from this graph is that there's a lot of variation among taxa. So the first point I want to make is that taxonomic groups vary in their response to climate change. Mostly in this particular group, they're heading northwards, and that's because we're only analysing the southerly distributed species that we'd expect to move northwards. Nonetheless, there's a lot of variation among the taxa, and that's one of the things that we've been investigating. Susie Mason, who may have gone to her talk yesterday, she's updated all these data, and one of the greatest things that she's done is she's now put error bars on all of those histograms, and that starts to show the degree to which species vary in their rate of response. So if you look now, just going back to the Lepidoptera, so about 155 species of butterflies and moths. So this is work that Georgina Palmer has been doing. So this is just the southerly distributed species, those species that reach a northern limit to their range in the UK, those that we'd expect to shift northwards. What's the variation within this one particular taxonomic group? So now we have the change in abundance. So this is from light traps and from UK BMS Transex. Then we have the change in the distribution size and then the shift in the range margin. So the moths are in white and the butterflies are hatched. And this is the 0 point here. So we're expecting most of these things to be increasing. And you can see that a lot of them are either heading northwards or increasing their area or increasing their population size. But you can also see there's a lot of variation, and slightly worrying here, quite a lot of things are actually decreasing when it comes to population size. So not only is there variation amongst taxonomic groups, there's also a lot of variation within spanning quite a wide range of differences. So another nice thing you can do with the Lepidoptera data is they're over such a long time period that you can start to split up that time period into two groups. So we have the earlier period and the later time period. So this is the 70s to the 90s, the earlier time period, and this is the 90s to the noughties, just to look to see if not only is there differences between taxa, differences within taxa, but is there variation within what an individual species is doing, and yes, there is. So the bottom line from all of this work that Louise Mayer did was actually only 5 out of these 37 species are showing qualitatively similar results. In other words, what they did in the earlier time period, they're also showing in the later time period. So there are some broad patterns that come out of that. One is that range margin shifts and distribution size increases were slightly larger in the second time period overall, and that abundance changes dropped in the second time period. But actually, there's lots of variation within species as well. And we looked to see what was the causes of that. So the top 3 plots here show what's going on in the earlier time period, and the bottom ones here show what's going on in the later time period. So 90s to noughties versus 70s to 90s, looking at habitat dispersal and abundance. And in the first time period, it's all being driven by habitat availability. So colonization is faster if there's more habitat availability for the species. So each dot here is a different butterfly species. But in the later time period, then it only seems to be the abundance trends that are an important predictor. So in other words, the main drivers and constraints of range changes and range responses are varying over time as well. So even more complexity to what's going on. And our explanation here is that actually you need to have a positive or stable population trend in order to be able to expand your range. And interestingly, dispersal never came out as being important. So I just want to finish off by saying something about sensitivity. So that's some new work that Georgina Palmer is looking at to see how species are sensitive to different climate variation. So we take the UK BMS data, or the light trap data from Rothamsted, and we fit those to models that contain a number of different climate variables for different seasons and for different years with lag effects, and run these models to see how well they fit the population trends. And we measure the sensitivity by the goodness of fit. So how well does the model fit the observed data? So for the comma here, we have about 50% of the year-to-year variation in abundance is explained by climate. So that's our measure of sensitivity. And then when you look across all of these different Lepidoptera to see what are the main drivers of those population dynamics, so from the models we take the top 3 climate variables, so whether that's temperature or rainfall, which season it's in, and whether it's a lagged effect or not, you see that there's a huge amount of variation in the rainfall and temperature variables, the climate variables that species are responding to. So what we have here along the bottom is the number of different Lepidoptera species that have any of these variables within their top 3 climate variables that fit to the model. So in other words, these are the most important variables that are driving the population variability. So a huge amount of change, not only in which variable it is, whether it's temperature or rainfall, and also whether it's a positive or negative response. So species change considerably and show considerable variation in their sensitivity. So if you just summarize that across the different Lepidoptera groups. So this is essentially the R-squared value from all those species-specific models where the climate variables have gone in, and we've looked to see how good that fit is. So you can see that there's quite a range of sensitivity. So the worst was where we had only about 10% of the variation was explained for a species that is not at all sensitive to climate, to another species that's actually very sensitive to climate, and about 90% of the year-to-year variation is explained by climate sensitivity. And as you can see here, what Georgina found that across all the Lepidoptera species, it's about 45%. So what we're now going on to do is to see, based on the sensitivity of species, we now want to know something about their exposure to the climate, how the climate has changed for them, for those particular parts of the climate that are important for that particular species. So just to finish then, where we know that species are responding to climate, but there's a lot of variation and a lot of different ways of doing that, with considerable variation between taxa, between species, and also within species over time. Lots of variation in species sensitivity to climate and to different aspects of the climate as well. And of course, it's very interesting to see that rainfall is quite important for a lot of Lepidoptera, and yet most of the results I've shown you is made the assumption that it's temperature. We're now going on to see what's the relative importance of a lot of these factors for species, whether it's habitat sensitivity, dispersal, and so on. And then, of course, what we're now starting to go on is to think a lot more about evolutionary changes and the degrees to which those might be important as well. So thank you for listening.
Thanks very much, Jane. We've got time for a very quick question if somebody has a quick question. Just very quickly then, Jane, you know you showed that graph, I think it was from the GCB paper with the different taxa. Has anybody, obviously, they move to different extents of range. Has anybody looked for sort of functional traits that underpin that? You know, whether mobility, for instance, and certain groups make some move more? Because I noticed fish are doing.
Yeah, no, so that's something that Georgina Palmer's looking into. But it's quite difficult to get good estimates for dispersal across the different taxonomic groups. Yeah. So even for butterflies, where you think there should be good bits of information for that, then mostly if we want to understand something about dispersal, we're taking a morphological measurement like wing size or body size or something like that. So that's probably the only life history trait that you might be able to.
Yeah, and probably hugely variable even with the taxa. Thank you very much, Jane, for that. Excellent. So it's a pleasure now to introduce our next speaker, Sabine Newton, who's again going to talk to us about a different approach for assessing how invertebrates are going to respond to climate change, and that's transplant experiments. Thanks, Sabine.
Thank you, Scott. So hello, everyone. Today I would like to introduce a very different method to study climate change impacts on species and communities, which is transplant experiments. And my co-author, Nigel Andrew, and I myself, we have conducted transplant experiments and recently written a book chapter on this topic. So as we all know, climate change is a major threat to species and communities, and according to the latest IPCC predictions, temperature will continue to rise according to the CO2 emission levels. Precipitation patterns will alter, and the frequency of severe weather events will increase. So with all these continuing changes of abiotic factors, it's now more urgent than ever to study climate change impacts on species and communities. So very briefly, climate change, as the increase in CO2 and temperature and altered precipitation patterns, impact species individualistically, and they can respond by changes in behavior, changes in their physiology, shifts in the timing of spring events and phenology, changes in their geographical distribution. And these individualistic responses can then lead to shifts in biotic interactions in terms of temporal and spatial mismatches between interacting partners. And this again can then lead to changes in community composition and structure with potentially far-reaching consequences for ecosystem functioning. So, well, how do we study climate change impacts? Well, there's a variety of ways and approaches to do this. Most commonly used are things like bioclimatic modeling, SDMs, or glasshouse experiments. Well, these are fine if you're looking at single species responses only, but if you want to look at community-level processes, you need to move on. So what many people have done is they use latitudinal or altitudinal gradients in field observation as a surrogate for climate change, or as a surrogate for climate. Or they did experimental warming in smaller plots in the field, which is on a rather smaller scale. Or what I will present here is you can do a field transplant experiment, which is a very powerful method because you can move species and organisms directly into a new climate and assess under natural conditions what's happening to them. So it's a very direct test what might happen in the future. So to do a transplant experiment, there are multiple ways to go about it. So if you look at the graph here, in white you can see the species range. And you can do different types of transplant experiments by moving species either latitudinal or altitudinal. Latitudinal is seen in panel A, and altitudinal is seen in panel B. And then you can choose, the black triangles are populations or individuals of species within their range, and they can basically be swapped reciprocally between a central and a peripheral area of their range, or they can basically be moved outside of their range, or as a third approach, you can source individuals or populations from species across multiple locations across their current range and then transplant them outside of their range. So there's a range of questions which people have been addressing when conducting transplant experiments, which are looking at ways how species can adapt to new climate and if range shifts are possible. They looked at changes in phenology and shifts in species interactions and communities, and they looked at genotypic and phenotypic responses and tried to disentangle them. So I don't want to go through all of them, but I will briefly introduce two questions to you, shifts in species interactions and communities, because I have conducted a transplant experiment looking at these two questions. So I did a multi-species transplant experiment where I moved plants to look at insect responses. And to do this, I transplanted 640 individuals of 8 plant species into 3 field sites. If you look at the map on the right-hand side, this is the distribution of the gray ellipse is the distribution of these 8 plant species. And we conducted the experiment in New South Wales, which is at the southeastern tip of Australia. And we transplanted into a control site, which is in the approximate center of the species distribution, and 600 kilometers north of the northernmost boundary, into plus 2.5 degrees warmer sites in mean annual temperature. And then, please note, this is Australia. You need to transplant north to be closer to the equator. And then we monitored insect colonization over 1 year, assessed herbivory, and characterized the insect community. So with this setup, we looked at one of the most fundamental species interactions, which is herbivory, and we were interested to find out if herbivory as total will increase and if herbivory damaged types will change in their relative distribution. So we did this experiment with 4 plant species, but I only will show the example of one species to illustrate the general trend. So total herbivory after 12 months in the field was very low, between 4 and 10% only, and we didn't find a consistent increase of herbivory at warm sites. We were there interested in the distribution of herbivore types, such as grazing, mining, sucking, and chewing, as you can see on the graph, which shows the relative distribution of these feeding types after 1 month, and they were relatively consistent between control and warm sites for all 4 plant species, which indicates to us that at least on these species, dominant feeding types such as sucking and chewing will be staying dominant in the future as well. So with the same setup, we were then interested in looking at shifts in herbivore insect communities, and we used 8 plant species for this experiment. And again, I only show the example for 1 plant species to illustrate the general trend. So we identified 273 beetle and bug species and classified them into feeding groups or feeding guilds according to their mouthpart morphology and targeted plant tissue. And on species level, we found that there was a more than 90% turnover, which indicated that species will colonize new host plants in the next decades to come. And when we were looking at feeding guild level, we, as you can see on the graph here, which shows the relative distribution of feeding guilds, such as leaf chewer, mesophyll feeder, and phloem feeder, we found that the general distribution of these feeding guilds was similar between control and warm sites for the 8 plant species. And which indicated that if species drop out, they will be replaced by other species in the same feeding guild. And interestingly, when we were looking at congeneric plant species at warm sites, we found that the feeding guild structure was very different. That indicated to us that feeding guild structure on those plants is driven by the host plant identity. So moving away from my studies to the general transplant literature. So we did a literature search in Scopus and Web of Science using the search terms climate change, transplant experiment, or translocation. And we found 69 studies, of which 22 only involved invertebrates. And we were then interested in trends and characteristics and what people did most by using that technique. And we did network analysis with Cytoscape. And what we could find is here we can see the relationship between continents and climatic drivers tested. So the nodes or circles in green are the continents, in yellow are the climatic driver, and the edges or lines between them are the transplant type. If you move along latitude in orange, longitude in brown, or altitude in blue. And the size of the node corresponds to the number of connections, and the thickness of the line corresponds to the number of studies. And please keep in mind there are very few studies we found overall. So we found on general trend that 3/4 of the studies were conducted in the Northern Hemisphere, mostly in North America and Europe. More than half of the studies looked at temperature. Very few studies actually looked at precipitation, and only 1/4 of the studies were conducted on the Southern Hemisphere in Australia or South America. So then we were interested in the relationship between transplant type and climatic driver tested. So again, the nodes are in gray, indicating the transplant type, in yellow, the climatic driver, and the lines are the organisms transplanted. In green is plants, in purple is insects, and in brown is soil cores. and the dashed lines are plants with soil cores attached to them. And what we found here is that most of the studies, about 2/3 of them, used latitude to assess temperature changes, and they transplanted or translocated plants and insects in similar manners. And very few studies actually translocated soil cores to look at invertebrates in the soil, and few studies looked at precipitation differences. So then we were wondering or asking ourselves what's missing. What did people not put or incorporate into transplant experiments, and what could be looked at? So people usually didn't look at microclimatic variation. So for example, in summertime, the temperature on the ground can heat up tremendously, and these changes cannot be picked up by local weather stations. So then people did also not look or incorporate behavioral or physiological adaptations if the insects on the plants, for example, move up or down to cool or to heat up. And then climate variability and severe weather events. So in summary, we take-home messages. We found that field transplant experiments are powerful tools to study climate change impacts, especially on communities. They are very useful to complement other approaches such as modeling and short-term experiments. And the majority of them were using latitudinal gradients to look at temperature changes on the northern hemisphere. And they largely focused on macroclimatic changes. And none of them incorporated microclimate or behavioral or physiological adaptations. And in the book chapter, we go into some suggestions for future transplant experiments and what could be incorporated in them. So thank you very much for listening.
Thanks very much, Sabine. So time for questions. Yeah, sure.
Thanks very much. I think the kinds of things that we're learning from these experiments that will be more and more interesting. So when you're planting plants beyond their current native range, they're going to be experiencing a mix of herbivores, some of which they already experienced in the native range and some of which are different, and not necessarily all of which are going to be the ones that the plants will experience in the future because they're in different conditions. So do you think these results are reflected in the feeding gills that are available at the new locations, or the plant's susceptibility to gills or to the individual species? Just trying to think how you really translate this data into the future for these plants within their current native range.
Yeah, we need to bear in mind that when you actually move the plants from one spot to another plot, you actually have an empty plant, which is not occupied by any insect. So it's basically a new assembly, which would never occur in nature because it's basically full of insects. So, and I think also the insects usually, there might be very new insects on these plants because none of the species we found at the control sites were actually at the warm sites. So I think that was kind of a little bit of an artificial assembly of a new community. But the striking thing was on functional guild level, that we could see that the guild distribution on a plant is basically characterized or driven by the whole plant identity. That was actually the main question to ask in this report.
And not the feeding gill that was actually available, not the pool of species that could have been yes.
Excellent. Thanks very much, Sabine. Okay, so we're bang on 2:00, so it's a real pleasure to introduce Professor Helen Roy, who's going to talk to us on encounters with aliens, insect invasions in a changing world.
Well, thank you very much, and thank you to Scott and Will for inviting me. And I apologise for my voice. It's an indication of quite how much I've enjoyed the conference. I'm not feeling unwell, just having talked far too much, I think. There are many, many people I could add to this slide as co-authors, and I apologise for not listing them all. This has been hugely collaborative work, and I will try and represent people as I go through, but for sure I'm likely to miss people, and I do apologise. So, well, the first thing I thought is that I'm going to change my title, and I'm going to pay a tribute to the culture of Liverpool. And it's been a great few days in Liverpool, and I thought, actually, it's a great song, and certainly it gives a sort of scenario on which you can all think as I go through my talk, and in terms of imagining all the insects sharing all the world, because that's what the sort of ultimate of invasion biology would perhaps lead to. So, as well, thinking back to the first talk of this afternoon and thinking about the challenges that we face and the things that we have done and the things that we haven't done, I think that we've talked a lot about the interactions between drivers of change, but we haven't done a lot of work around the interactions of drivers of change. We've done a lot about thinking about rankings, so which is the biggest threat to biodiversity, but I think we need to take them in their totality and begin to think more cleverly about bringing them all together and thinking across these drivers. And it's quite a while ago that climate invasion were described as a deadly duo, and this was published at a conventional biological diversity meeting. And we can certainly see that over time there's an increase in the number of species arriving. Here's a pointer, yeah, so we can see this increase over time, and this is for Europe. And I'm going to interchange between using the words non-native and alien, and I apologize for that, but essentially what I'm referring to are species that have been introduced in one way or another by humans. And I'm only going to use the word invasive when I'm talking about that small proportion, that small fraction that actually have been shown to have some demonstrable biodiversity impact or perhaps an economic or societal impact. And we know that those impacts for that small proportion of species can be extremely costly, and the estimated direct cost for Europe was 12 billion euros. But we know that there are many, many different interacting factors, and we know that globalization and environmental change are facilitating invasion. And these particular maps over here on the left-hand side are from a paper that we've now got in revision, we just heard in the last few days. It's led by some fantastic modelling work that Dan Chapman has been doing in CEH Edinburgh, looking at agricultural trade flow and how that's influencing the spread and movement of non-native species. And then there's been some really wonderful work by Regan and her collaborators and her team, and she's been looking at how these different drivers, these different factors have varying importance throughout the invasion process. And I think this is a really very nice paper in terms of taking a global perspective and looking at the threats from invasive alien species alongside other factors. And we can see that there are some factors that are very important in terms of introduction, of course, things such as trade and the movement through airports and seaports, etc. And then we can see that in terms of establishment, there's a whole other set of factors that we need to consider, and climate, for instance, would be certainly important there. So I just thought I would mention some of the work that we've been doing at the Centre for Ecology and Hydrology. And one of the projects that I lead is a DEFRA-funded project, the GB Non-Native Species Information Portal, where we gather all of the information that we possibly can on non-native species within Britain. And we have about 2,000 established non-native species in Britain. So these are the species that are sustaining populations within Britain. They're reproducing or sustaining in one way or another. And every year we produce a scorecard for Great Britain. So this is the latest scorecard. And we can see that the plants are numerically dominant with about 1,500 established non-native plants and a much, much smaller number of established non-native animals. But then if we look at the impact, and we need to get better at quantifying impact, so there's a little bit of error probably within this, but we can see that for sure the non-native animals are those that we should be perhaps more concerned about, with about a third of them having some demonstrable impact. And I just thought I'd break down those non-native animals to see the dominance of invertebrates within that, and the number of insects too, with about 241 established species of non-native insect in Britain. And if we have a look at invertebrate invasions over the time, so this is now GB data, and we look at the cumulative number of invertebrates over time, we can see that it follows a very similar pattern to that when we put together all the species and when we look at the trends across Europe, for instance. And indeed, you could plot this kind of figure anywhere in the world, and you would see this escalation. And there's some wonderful work that's coming out from a paper that Hanno Siebens has been leading with a number of us in the room as co-authors, looking at these accumulation curves over time. And it's really fascinating but quite worrying to see that there isn't any slowing in terms of the numbers of new arrivals all around the world. And I just thought it was quite nice to add a few pictures of some of the species that we're seeing from the sort of cockroaches, the first record that we have in the GB database. And then some more very recent new arrivals, including Vespa velutina, which was recorded in Gloucestershire just in September, hopefully has been eradicated, but it's really important that anyone's on the lookout over the next year, and certainly February onwards. So it's all very well documenting all of these species, and we can produce some really beautiful pictures and trends, but actually, in terms of doing something about the ones that are causing some kind of biodiversity impact, can we make predictions? So perhaps we could prevent their arrival in the first place, or at least be well prepared for their arrival so that we can do something when they do arrive. And we carried out a horizon scanning exercise for Britain, and we borrowed methods that are probably widely known that Bill Sutherland uses to generate, for example, the top 100 ecological questions. So we use these consensus methods where you gather experts together, and it can be tremendously valuable when you've got quite a patchy data set. You've got a lot of unknowns in that data set, but you can draw on expert opinion to fill in some of those gaps. But you can also use the best available evidence that you have through the literature or through risk assessments to pull together lists. And we very ambitiously looked across terrestrial, marine and freshwater systems, and across a whole variety of taxa, pretty much only excluding microbes. So we had all these groups working together and coming up with their lists, and then we came together as a group of experts to rank the list, to bring the list together and to rank all of our species, and to look at what are the ones that we should be really worried about in the next 10 years that are going to pose a threat to biodiversity within Britain. We published this in Global Change Biology, and I don't know if it's a measure of success or if I should be standing here in tears, but 7 out of our top 10 species have now arrived. We only published the paper in 2014. So maybe actually 10 years wasn't enough. Maybe we need to be looking further forward so we can actually have something in place to be able to prevent the arrival, for instance, to do something a bit longer, to make some kind of plans around these lists that we generate. But nevertheless, perhaps it does demonstrate that the method was working. And certainly one of the factors that we brought into the consideration was thinking about things such as climate. So, for example, the Argentine ant was not in our top 10 set of species. It was ranked between 11 and 20. And that was because we need the climate to be a little bit warmer before that species can take off in Britain. But for sure, it's not going to need to be very much warmer, and we'll start to see that species more frequently. So I'm just going to be a little bit indulgent, and perhaps again with a sort of Liverpool reference, beetles are very appropriate. And so I'm going to bring in a ladybird perspective. And I think Will was very generous with my abstract. He knows that this was going to be the case. So I run the UK Ladybird Survey, which is essentially a citizen science project, but is standing on the shoulders of giants. And Mike Majuris, I ran the survey at first with Mike Majuris, who sadly died in 2009. And I guess this is a bit of a commercial break as well, that he had a manuscript, a book that he had written that was almost ready for publication when he sadly died. And Cambridge University Press asked myself and Peter Brown if we would bring it up to date so that it could be published. And it was published a couple of weeks ago. It's on the stand of Cambridge University Press. And I'm sure if you're looking for a Christmas gift idea, then, well, I could recommend A Natural History of Ladybird Beetles. So I'm standing on the shoulders of these people who have led the survey before me, and we have these amazing data sets, just in a way as there are for the butterflies. Of course, it's not quite such an impressive data set, but nevertheless, they're pretty good. And one species in particular, Harmonia axyridis, has really exploded interest within the ladybird world. And this species arrived or was first recorded in the UK in 2004. It's likely that there was one record from the year before that. And Mike put out a press release when it arrived and said, this is a new ladybird that has arrived in Britain, but not just any ladybird. It's Harmonia axyridis, the most invasive ladybird on Earth. So if you want to find out more about the Ladybird Survey, you can visit our website. And I will also say a few of these slides now that I've put in are just to show you some beautiful pictures of beetles. But there are many, many different ways that people contribute records to the Ladybird Survey. So we don't have a systematic translate method as they do with the butterflies, for instance. It is just ad hoc occurrence data. But we use iRecord for gathering that data. People can download a free smartphone app. We get lots of letters still coming through the post, spreadsheets, emails, lots and lots of different ways in which we gather data. And we've now got more than 200,000 records within our database, and our oldest record goes back to the 1800s. So it's actually fantastic for us in terms of not only looking at generally at environmental change, but particularly looking at certain markers, such as the arrival of Harmonia axyridis, and what is that doing to the distribution of species over time. And we can see that records over time, it's looking really very healthy for the ladybird survey, and hopefully we're going to go from strength to strength in terms of gathering more and more records. And it can kind of look a little bit bleak in the early days from when the Biological Records Centre was established in the 1960s and began to host the ladybird survey and alongside a number of other schemes. But really, when you think about it, it's really the online technology that has allowed us to expand and get more people involved and more records coming in. And for sure, this is going to be fantastic for us in terms of gathering the data that we need to address these big ecological questions. And what's really inspiring for me is that within that database, we now have nearly 19,000 names of recorders. And that we have many, many thousands of people contributing ladybird data and getting really interested and passionate about ladybirds. So hopefully, yeah, Tom's map is working. There we are, Tom. So this is Tom August produced this beautiful map for us, and it just shows this really impressive invasion and the way in which the harlequin ladybird spread at 100 kilometers per year. And what you can see of this flush of red, and if you look at the time, it's often in the autumn time or the spring emergence of the harlequin ladybirds, but also the autumn invasion into people's houses. And this autumn was a particularly spectacular invasion into people's houses. But I think it's a really fantastic visualization of how the data's come in. And it has to be one of the best invasion data sets for a particular species, and it's down to all of the many, many people who've diligently sent in their data over time. And we've been able to use that data to look at declines in native ladybirds, a little bit depressing, but it's great to be able to use the data to look at impacts and look at impacts empirically and look at, it's correlative studies, but looking at changes in distribution in response to the arrival of Harmonia axyridis. And I won't go through this whole panel, but just with the two-spot ladybird in the top corner there, you can see the solid line is without the harlequin ladybird in Britain, and the dashed line is for the squares with the harlequin ladybird. And we can see this 44% decline in the distribution of two-spot ladybirds, strongly correlated with the arrival of Harmonia axyridis. But we can also then use traits of the ladybirds and also bring in climate data as well to begin to look at climate and invasion. And this is some work from my PhD student, Richard Comont, who graduated a few years ago. And he brought in these different layers within the models and showed very strongly that there is a climate effect, and that climate effect varies between one species and another. There's also effects of habitat too, but the overriding factor that at the moment is influencing the distribution of ladybirds in Britain is Harmonia axyridis, the harlequin ladybird. So we've summarized all the information that we've been gathering over the last 10 years, and a lot of it has been based around this survey data where we've been getting people sending in their records, and we produced this paper from Ecological Entomology earlier this year. We've still got an awful lot of unanswered questions. There's still a lot of work to be done if you're interested in joining the field. We've been going global. We've been now working with people in Chile who are running citizen science projects and looking at the ecology of Harmonia axyridis there, and also with people in the Czech Republic and also Slovakia. And we produced a paper on the global distribution of Harmonia axyridis and looked at variations in its ecology around the world. And it's really fascinating how it does vary in one place to another. Of course, that adds another layer of complexity for us. So these amazingly charismatic beetles and all of these incredibly inspiring recorders have allowed us to begin to unravel the ecology of Harmonia axyridis. We have a long way to go, and I think that understanding interactions, understanding the different interactions between different drives of change is certainly going to be the next things that I'm beginning to focus on. I also want to begin to think more about citizen science and alien species and think about it in other places, and had the privilege with the British Ecological Society of running a workshop in East Africa to look at the opportunities and challenges of expanding citizen science work into East Africa. So thank you all very much for listening. Thank you to all my collaborators, the Ladybird Survey as well, and various funders included too. Thank you.
Thanks very much, Helen. We've maybe got time for a very quick question whilst people move around. Quickly, so I can imagine how climate change facilitates invasions by making a habitat warmer, for instance. So, you gave the example there of the Argentine ant. Do you think that climate change also can weaken natives? By their very nature, they're less adaptable than invasives.
Yeah, I think that's a really excellent question. I think when we're beginning to embrace some of the complexity around this, we need to think about how these different drives of change are stressing perhaps the native species in this case, and then, for example, allowing invasion to occur. And we know that there's going to be changes in terms of interactions between pathogens and insects, for instance, and how does that then influence the interactions and the ability of other species to then arrive. I do think the challenge is that we've got to get more complex, and we've got to start thinking about networks and multiple drives of change. And to me, that's really exciting. But we have a long way to go. But yeah, I agree. Yeah.
Thank you.
Thank you.
Okay, so we'll move to our next speaker now, James Riles from Western Sydney University, who's going to speak to us on silicon effects on insect food web dynamics and elevated CO2.
All right, thanks, Scott. And thanks, everyone, for being here. It's a pleasure to be here. And joining in the excitement of silicon research. So I'm going to talk to you today about a BES-funded project looking at the effects of silicon and CO2 on multitrophic interactions, which it's part of the interim between it was a project that I did in the interim between finishing my PhD and starting my postdoc position at the Hawkesbury Institute for the Environment at Western Sydney Uni. So first of all, we live in a green world. Despite 25% of species being herbivorous insects, we live on a green planet that's dominated by terrestrial plants. And this is because plants are very good at protecting themselves. And they can protect themselves via both top-down and bottom-up mechanisms. And plant chemical defenses in particular can restrict damage caused by insect herbivores both directly by, for example, being toxic to the herbivore, or indirectly by influencing the third trophic level, so for example by recruiting natural enemies. And although these top-down and bottom-up mechanisms generally protect plants, plant chemical defenses and other defenses can actually weaken top-down control of herbivores by reducing the quality of the herbivore for its natural enemy. So this project focuses on mainly on silicon defenses in grasses and how this affects the herbivore and its predator. And silicon itself is super important. It's the second most abundant element in the Earth's crust after oxygen. It's been known for many years to increase the general growth and yield of plants. And many plants have different accumulation capacities. So some are active uptakers, some are passive uptakers, and then others don't take up silicon at all. But grasses are among the best active uptakers of silicon around. And aside from the general increases in growth and yield, there's a number of other benefits of silicon. So they've been shown to alleviate drought stress, heat stress, salinity, things like metal toxicity. But what I'm really interested in is the ability of silicon to defend against insect herbivore pests. So here are some scanning electron microscope images taken from Sue Hartley's lab, which show how silicon, which is taken up in the form of monosilicic acid, can actually be deposited as these phytoliths or silica bodies on the surface of the plant. And these are really quite unappetizing for the insect herbivore. They can wear down their mandibles, reduce nutrient acquisition via reduced digestibility and palatability. And while they've been known to be a really important anti-herbivore defense, their role higher up the food chain has rarely been tested. And then on top of that, we've got climate change, especially changes in CO2 concentrations in the atmosphere. And it's been hypothesized that back in the Miocene period, when CO2 concentrations were extremely low, this was about 20 million years ago or so, and the CO2 concentrations were about 200, 250 parts per million. The plants fixed less carbon, which meant that carbon was substituted for silicon as a cheaper structural alternative. And this actually prompted the evolution of high silicon accumulating grass species. But what's going to happen in the future? What's going to happen when CO2 concentrations increase? Well, under elevated CO2, we may well see this decrease in silicon uptake as carbon availability increases. So we may see this decrease in the defensive capability of the plants, the silicon-based defense. And so in order to test this, we used 3 plant growth chambers with 3 carbon dioxide concentrations. 550 parts per million is predicted for the around mid-century, and then 800 for the end of the century. And we grew plants, specifically harding grass, Phalaris aquatica, in these chambers, which is a naturalized common Australian grass species. It's normally used for pasture and fodder. And then in each chamber, the grasses were grown in each chamber, and then half of the grasses in each chamber were supplemented with silicon and half of them weren't. I'm not going to go into too much detail about it because I want to get to the results, but essentially we did 2 feeding trials. The first one involved looking at the effects of feeding the grasses in the different conditions to crickets and using controls to account for the water loss. And then recording things like change in cricket mass, the amount of mass eaten, frass production, and we calculated things like approximate digestibility. And then the second feeding trial involved feeding the crickets to the praying mantids. So the crickets were fed for 2 weeks on grasses in the different conditions, were fed to praying mantids, and we recorded things like change in mantid mass, the mass eaten, frass production, things like that. So it's a really simple experiment. And then we took some plant samples as well to measure plant characteristics. And the results showed that silicon supplementation increased the total grass mass, so it had a fertilizing effect on the grass. Expectedly, we saw a increase in silicon concentrations under silicon supplementation treatments. And then we saw this decrease in silicon concentrations under elevated CO2, which was also shown by the negative correlation between silicon and carbon concentrations in the foliage. So we saw this sort of trade-off between silicon and carbon, which was really cool to see. The results from the herbivore feeding trial showed that when crickets were fed silicon fertilized grasses or silicon supplemented grasses, the crickets ate less grass, they lost more weight, and they produced less frass than when they were fed on control grasses. And we didn't see any CO2 effects, so the results were really simple. We only saw a silicon supplementation effect. And then silicon concentrations were negatively correlated with the amount of grass eaten, which probably wasn't due to digestibility because we saw this decrease in frass production and no compensatory feeding or anything. So in fact, we actually calculated approximate digestibility by subtracting the frass production from the grass eaten, and then using that to calculate the proportion of the grass eaten, which wasn't significantly different between the control and silicon supplemented treatments. So it's probably more related to simply the crickets avoiding the grass due to the nature of the grass, the unpalatability of it. So results from the predator response feeding trial showed similar results to the cricket. So when the praying mantids were fed on crickets that themselves had been fed on silicon supplemented grasses, the mantids ate less crickets, they gained less weight, and they produced less frass than those that were given control crickets. And then again, we saw no CO2 effects. So here's a summary of the results. Well, we saw this silicon-carbon trade-off, and where silicon decreased under elevated CO2 concentrations. This was obviously not sufficient or not large enough to affect the herbivore or its predator. Silicon supplementation reduced feeding by crickets, most likely because it avoided the tough or abrasive tissue. And this subsequently reduced the feeding performance of the praying mantid predator. So in other words, silicon supplementation, while it reduced herbivore performance and increased bottom-up control of the herbivore, it actually decreased top-down control of the herbivore by the natural enemy, by the predator. and whether this will have a more pronounced effect in other species with other grass species with higher silicon accumulation capacities or other insect species, I don't know. But in this case, the effects of silicon addition were not likely to be diminished under future climate change scenarios. So understanding how plants accumulate silicon, how they invest in these silicon and non-silicon based defenses, and how it can be driven up the food chain to affect these predators will be important in the future. And then this is going to follow on to my project now, looking at called Get Tough, Get Toxic, or Get a Bodyguard, which refers to the plant's ability to defend itself through toughness, toxicity, or by recruiting natural enemies. And then I'll be doing a number of experiments looking at silicon effects on plant characteristics and below-ground herbivory using rain exclusion shelters and field warming study. And then I'll be looking at the effects of plant parasitic silicon on plant parasitic nematodes in the glasshouse experiments. So if you want to keep updated with that or want to see what I've done previously, please check out my website. And on that note, I'd like to thank my supervisors, Scott and Sue, for supervising the project, and then Andrew and Goran for providing technical assistance. And then I'd also like to thank the BES and HIE for funding the research, and thank you all for listening.
Okay, so we've got quite a little bit of time for questions. Anybody has any? Yep.
Could be a factor. I mean, in this scenario, the predator is probably less honed in on those kind of olfactory cues, but probably not likely to have an effect. But I would definitely advise using sort of whole plants instead of cutting plants because it could affect, especially with other predators that are more honed in on those kind of cues. Yeah, good question.
Yeah, Helen.
I'm really fascinated by this environment, and it's possibly not a question, but I just wondered if you thought any consideration around endless environments and how they might affect in terms of medium term some of the work.
Yeah, I mean, I certainly think it would be an interesting thing to look at. I haven't actually thought about it, but I have now.
I didn't know about in Auditorium. I didn't know anything about endless environments in Auditorium, so but certainly this has pop up in one pilot.
Yeah. No, certainly. I mean, it'd be quite likely to have a knock-on effect on them as well. Yeah. I suppose so.
Is there another question? Yeah.
Yes.
So you might expect less of a response from the CO2 and silicon with C4.
Potentially, yes. I'm not entirely sure. There have been some studies looking at C3, C4 comparisons. I'm not sure. I'm not too sure about what.
Okay. Well, I was a bit concerned when James said he wanted to become more interested in silicon implants, but it seems to have paid off. So thanks very much, James. Okay, so now it's a pleasure to introduce our next speaker, Paul Parham, who's actually at the University of Liverpool, but based in London, I think, at Imperial. And he's going to speak to us on the optimal temperature for mosquito-borne disease transmission.
Thank you very much. Thank you to Scott and to Will for inviting me to speak today. So what I want to talk about today, just for 10 or 15 minutes, is sort of some work in progress, I suppose, on work I've been doing recently on mosquito-borne diseases. So I've been interested in mosquito-borne diseases now for sort of 10 years or so, and particularly the response to potential mosquito-borne diseases to climate change. And of course, with the sort of theme of this work, with this particular session around which little things will run the world, you only need to look at the recent Zika outbreaks to know that mosquito-borne diseases still pose a huge threat to humans, killing around 1 million or so individuals a year. So mosquito-borne diseases have caused a huge burden, both in terms of mortality and morbidity, for a while now, and are likely to continue to do so. So what I want to talk about in this session is trying to sort of untangle, I suppose, to some extent, what temperature we might expect some of these diseases to be worse at. So the context of this study, a few years ago there was a paper published in Ecology Letters looking at what temperature malaria might peak at. And the conclusion from that study was that malaria seems to peak at around 25 degrees C. It's a unimodal curve, so either side of that peak, malaria causes less of a burden to humans. And one of the interesting conclusions from that paper was that that peak transmission temperature was around 6 degrees lower than we previously thought. So this was an interesting result, largely because that sort of 31 degree peak was something that we found in a paper a year or two before that. So the graph at the bottom of that slide sort of illustrates their main result. So in the red curve here was a paper that we published in 2010, showing that one of the key metrics of malaria transmission peaked around sort of 31 degrees C. Whereas this paper was predicting that malaria was peaking closer to around 25 degrees C. So this was an interesting study that kind of got me thinking about some of the reasons why these results might be slightly different, and in sort of a sort of wider picture, I suppose, what temperatures could we expect different diseases to peak at? So does malaria have a different transmission peak to something like dengue or chikungunya or Zika virus? So this was something that was a sort of focus of this work. One other thing to note about this paper was that the authors used this thing called the entomological inoculation rate, which I'll say something about in just a couple of slides' time, whereas we'd used this concept called the basic reproduction number. Now, my background is in infectious disease epidemiology, particularly more on the mathematical epidemiology side, and the basic reproduction number essentially quantifies the number of secondary cases that you will get per primary infection. So, for example, if I've got flu today and nobody else in this room has got flu, by the time I recover, if I've infected two of you through conversations that we have after this talk, the basic reproduction number for that disease in this population would be 2. Okay, so this is infectious disease epidemiologists spend a lot of time trying to estimate what this basic reproduction number is, because it tells us something about how severe the disease is, how transmissible it is, and also how hard it is to control. So I was interested in whether or not, because this paper essentially compared two different things, we'd looked at what temperature the basic reproduction number peaks at. They'd looked at what temperature the entomological inoculation rate peaks at, whether that is actually important. So the starting point for this was, I'm sure some of you are quite familiar with this, is what's called the Ross-McDonald model, which has been around for a very long time now, 70, 80 years or so perhaps. And the Ross-McDonald model essentially underpinned almost all mosquito-borne disease modelling. So whether you're modelling malaria or dengue or Zika, you tend to start from the Ross-McDonald set of assumptions. I've put the equations down in the model. It doesn't really matter too much what they mean, but essentially what you do is you take the mosquito population, you divide it up into a set of infection states, and you also take the human population and divide that up into a few different infection states, and you model the transmission between the two. So the Ross-McDonald model essentially does two things. It tracks how the number of infected humans changes over time, So this essentially is the prevalence, let's say, of malaria in a population over time. And this the second equation here will model how the prevalence of malaria, let's say, will change in mosquitoes over time. And there's various parameters that will represent that model. I won't say too much more unless people are particularly interested, but essentially just to say that this is a starting point for 80-90% of models that look at mosquito-borne diseases, Zika included. The problem you've got, I suppose, is that many of these parameters here depend on climatic variables, temperature, humidity, and rainfall. The scale's just slightly got cut off here, but this is a plot of biting rate of mosquitoes as a function of temperature. And again, I won't say too much about these particular parameterizations, but you can see very quickly that there are different ways of parameterizing different parameters in this model. They all respond differently to temperature, and different species have different temperature responses as well. Similarly, if I look at the survival of different types of mosquitoes to temperature, you can see these response curves are also very different. And you can also see that for different parasites, the rate at which they develop inside the mosquitoes also varies quite considerably with temperature as well. So here's a couple of examples. So Plasmodium falciparum and Plasmodium vivax, two of the most common types of malaria, their temperature response also varies quite importantly as well across this range. So when you put all that into the mix, it's not obvious to find out where these diseases might be worse and therefore what the potential impact of climate change might be to some of these types of diseases. The other problem you've got with these kind of models is there's various different risk metrics that you could use. So what does it actually mean to say malaria peaks at a certain temperature? You need to be a little bit more specific about the metrics that you're talking about. So as I mentioned, arguably the most important one is this concept of the basic reproduction number. It tells you about the number of secondary cases per primary case. And you can define the basic reproduction number R0 in terms of some of the model parameters. Again, it doesn't really matter where this expression comes from, but really just to make the point that you can derive the basic reproduction number from your model equations. However, you can also define a different set of metrics as well. There's something called the epidemic potential, which is kind of important as well. The reason the epidemic potential is important is because it sort of scales out, I suppose, the dependence on the vector model. So here, little m is the number of adult mosquitoes per human. Okay, now that, of course, is going to change with temperature quite a lot as well. Vector population dynamics change quite considerably with temperature and rainfall. So as the vector population dynamics change, the infectiousness or the transmissibility of the disease will also change quite a lot as well. However, the epidemic potential doesn't depend on the vector population dynamics, so it's an interesting starting point. There's a couple of other ones as well, as you might expect. There's what we call vectorial capacity. There's something called the entomological inoculation rate, and there's something called the parasite rate as well. Again, it doesn't matter too much what these different metrics are, except to say that they are different. You can define all of them from the model. So again, which one should we be looking at? If I want to know what temperature Zika is going to peak at, or malaria is going to peak at, and therefore what are the prospects for these diseases under different climate change scenarios, I need to define very carefully what I mean by risk. So what I was trying to do with this work is a few things, really. Firstly, determine what temperatures these different metrics might peak at, or do they all peak at the same temperatures, therefore it doesn't matter what risk metric I'm talking about. To investigate how they might vary from between mosquito pathogen systems. So for malaria, for example, there's about 40 or 50 different types of Anopheles mosquito that can transmit malaria. So how does it vary by vector pathogen system? Lot of uncertainty in terms of the data, the empirical data that goes into parameterizing these kind of models. So what effect does that parameter uncertainty have on our predictions for where these diseases might get better or worse under different climate change scenarios? And ultimately try and tie all this back in, I suppose, with this more broad question about what climate change might do to certain diseases. I won't go through the top bit in too much detail, but except basically to say that you can do a fair amount of analysis on this system, a fair amount of analytical work, and you can show pretty quickly that these metrics do not peak at the same temperature. So the basic reproduction number will peak at a different temperature to the entomological inoculation rate, which will peak at a different temperature to vectorial capacity. So that's really, really important in terms of trying to work out where these diseases peak and what they might do next as temperature temperatures change. So the table at the bottom here is a sort of pairwise comparison of these different metrics. Again, I'm not going to go through the table in loads and loads of detail, except to make a couple of points. Firstly, the point that I just made a second ago, which is that in general they don't peak at the same temperature. Secondly, in quite a lot of cases, these sort of pairwise comparisons will actually depend on how you model the mosquito population. And actually how you model mosquito population can actually influence where these diseases peak by perhaps 2 or 3 degrees. So it's kind of important how you choose to set up your vector model. It does matter for your disease predictions. And the third thing it often depends on as well is not just the vector population, but a couple of parameters that are kind of important. The first is what we call the biting rate, essentially, and secondly, the survival probability of these adult female mosquitoes. Okay, so put all this together, it's quite... it's not a straightforward question to answer. Where do these diseases peak? So that's some general insights, and this table actually holds for any mosquito-borne disease. So again, whether you're talking about malaria or you're interested in dengue or you're interested in Zika virus, where these different metrics peak will depend on some of these factors. So just to illustrate this with a particular example, here's some numerical results from analysis that I've been doing around one of the key vectors of malaria in sub-Saharan Africa, namely Anopheles gambiae. And if you look at the transmission of Plasmodium falciparum by Anopheles gambiae, if you look at all the different parameterizations that you've got, you can look at where these diseases, where this particular system has its peak temperature, so around 29 degrees C for this thing that I called epidemic potential. Slightly higher for Plasmodium vivax, transmitted by the same vector. And in the table at the bottom here, what I've done is I've compared these different risk metrics according to a few things. So this is epidemic potential, this is the basic reproduction number, and this is the entomological inoculation rate. All different risk metrics for whether or not this disease might get better or worse under climate change. And I've done it for the two main strains, I suppose, of malaria, Plasmodium falciparum and Plasmodium vivax. And what I've done is I've actually included 2 different ways, I suppose, of modelling the mosquito population. This was a model that I developed about 5 or 6 years ago. In the Mordecai paper, this was the paper that I introduced right in the very first slide, that sort of came up with some revised estimates based on what we did. They took our model and modified it slightly. So a couple of things to note on that. Firstly, the sort of trivial observation, I suppose, that Plasmodium vivax peaks at a slightly higher temperature than does Plasmodium falciparum. Secondly, for a given system, so this is all for Anopheles gambiae transmitting Plasmodium falciparum, malaria. If I look at the entomological inoculation rate, these things differ by about 3 degrees C, so i.e. Where this sort of malaria peaks, as it were. So if I choose the vector population model from this paper, I get a peak temperature of around 26 degrees C. If I take the model that we developed about 5 or 6 years ago, we get 29 degrees C. And that's a huge difference, and that difference in temperature really, really matters. So holding all other parameters the same, the only difference between these figures is how you model the mosquito population. That can cause a huge difference in terms of your estimates for where malaria peaks. What about these 2 columns? You can see that if I look down the Plasmodium falciparum under one model, Plasmodium falciparum under the original model, again, slightly different in terms of where that prediction is going to be. If I look across for the basic reproduction number, again, actually not too different. So actually those difference in curves that I showed you right at the start is almost certainly down to how they parameterize their model slightly differently to the way that we did. So it kind of emphasizing the importance of model parameterization. So this, as I mentioned, is kind of work in progress. So some of the general conclusions that we can make already. The optimum temperature for different risk metrics will differ. So if you're trying to answer the question, where will these different diseases peak in terms of temperature, you need to be more specific about how you're quantifying risk. The other thing I didn't really note as I was going through this was that vector competence what you assume for vector competence actually matters in terms of whether or not you assume it has a temperature dependence. If anyone's interested in that, I'll say something more about that afterwards. How you model the vector population is important because that can affect where your peak is, and the results can be extremely sensitive to model parameterization. So we're doing this for a whole set of different systems, for transmission of dengue, for Zika, chikungunya, for malaria. And actually what you find when you start digging into the literature is there's actually very poor empirical data generally on different mosquito species. So this is actually kind of, although it seems like a very straightforward question, it's kind of hard to answer given the data that we've got. Relative humidity doesn't tend to play a big role in where that peak is, which is kind of reassuring because we have very poor data on it. And in terms of the malaria example that I just showed you, Plasmodium vivax peaks just slightly higher temperatures than does Plasmodium falciparum. Lovely, thank you.
Thanks, Paul. We're just out of time, actually, there, so, but we're very close to the coffee break, so...
I'll take questions. Yeah, of course.
That would be great.
Thank you.
Thank you, I appreciate that. Okay, I'll just give people a chance to come in. Okay, so it's an enormous pleasure to introduce our final speaker today, Rob Thomas from Cardiff University, and his talk is Grubs Up, Impacts of Climate Change on Invertebrates as Food for Vertebrates.
Thank you very much, and congratulations on lasting till the end of the conference. I have to confess to feeling a slight imposter in this session because my primary interest in invertebrates is in their role as bird food, and apologies for that. Now, the world is changing. We've spent a whole conference learning all sorts of reasons why we understand that the world is changing. And in the end, it's very important for us as ecologists to be concerned about the mechanisms underlying those ecological changes that we're observing all around us. So mechanisms are what I'm going to be focusing on, and invertebrates as a key part of those mechanisms help us to understand the recent changes that are being described, enable us to predict future changes, and crucially give us insights into the methods, the most effective methods of conservation of biodiversity into the future. So I'm going to be focusing on invertebrates as the key trophic mechanisms mediating the impacts of climate change, in particular on vertebrates. Now, I'll look at various classes of mechanisms, and one of the primary, perhaps most direct class of mechanisms is in invertebrates as food for vertebrates, mediating changes in the abundance of vertebrates. And a classic example is this endangered species of bird of prey, the Everglades snail kite, which feeds on these apple snails, whose growth and reproduction and population size is quite strongly temperature dependent. And within the range of current temperature variability, there's a strong positive relationship there. As temperature increases, snail abundance increases. And so within the range of current variation, extra warmth is very good for these snail kites who have a higher food availability, and their population responds positively to temperature following warm years. As temperature continues to increase, though, of course, ultimately that relationship will become negative. And so as we project climate predictions into the future, then we can see that ultimately water temperature will go beyond the optimum for this prey species. And so this will be very bad news, both for the snails and for their predators, the snail kites. But of course, it's not just temperature that is important when we're looking at climate. Other aspects of climate can be just as important, and in some cases, much more important than temperature. A nice example is the impact of rainfall on availability of earthworms as food for Eurasian badgers. And the behavior of earthworms is such, as we all know, in drought conditions, the earthworms go down deeper into the soil profile and become less available as prey for badgers. And this is important both for adult badgers, but especially so for badger cubs, who all are infected with an endemic protozoan gut parasite. And in years of low rainfall, those badger cubs are food stressed. They have a relatively low immunity to this gut parasite, and so they have a very poor survival. And so this is an effect of rainfall on earthworm abundance that impacts the demography of badgers via the gut parasites of the cubs. And as a result, badger abundance and distribution on a Europe-wide scale is rather more sensitive to the rainfall variable than it is to temperature variability. Of course, ultimately, changes in abundance of predators can lead to local extinctions. And a nice set of studies focuses on these Scoloporus lizards, which are invertebrate foragers. And in one study, which looked at 200 local populations, 12% of those local populations went extinct locally over the study period. And it turned out that those sites which had a greater increase in temperature over the study period had a significantly higher rate of local extinction of the lizard populations. And this was turned out to be mediated by the lizards foraging on invertebrates, but in a rather subtle way. And at high temperatures, the lizards were forced to spend more time sheltering in the middle of the day from the excessive heat. And this was traded off against the time that they were then able to spend foraging. So high temperatures, perhaps slightly counterintuitively, reduced fecundity and recruitment because those lizards were sheltering from midday heat for much longer periods and had a lower food intake rate, resulting for many of these populations in a local extinction. It's harder to put your finger on global extinctions of vertebrates mediated by invertebrates. An example that's often conjured up is the extinction of the golden toad. And there are, of course, a host of possible drivers of that extinction, some of which are climate-related, influencing food availability in various ways, but there are also potentially direct impacts of climate that extinction doesn't necessarily clearly show that extinction is mediated by invertebrate food supply. And of course, there are other aspects of that golden toad extinction that are rather independent of invertebrates as mediators of the population change. And in fact, this perhaps illustrates that often we should see climate change impacts not in isolation, but part of what you could think of as a deadly cocktail of both natural and anthropogenic changes in the environment. But it's not just population sizes that are changing. As we all know, there are big changes in distribution, and there are some nice examples of these geographical range shifts being mediated by invertebrates. One example, local to the UK, is sardines and anchovies are currently expanding northwards into the North Sea. And these are small pelagic fish whose the young fish feed extensively on relatively small calinous copepods. And there's a substantial shift in the abundance and distribution of two closely related species. Calinus helgolandicus is a small warm water species which is moving northwards and replacing Calinus finmarchicus, which is a rather larger cold water species, which is less suitable as food for sardines and anchovies compared to the smaller warm water species. So as Helgolandicus is moving northwards, then the small pelagic fish appear to be tracking that geographical range shift of their prey. Changes in distribution are not only north-south changes, of course. There are also altitudinal range shifts of vertebrates which are mediated by the impact of climate on invertebrates as prey. A nice example from the UK is that of golden plovers, a shorebird, which feeds on tipilids, crane flies, and the abundance and the synchronicity of crane fly emergence in summer is strongly dependent on low August temperatures the year before. And plover's distribution is moving uphill altitudinally, tracking lower August temperatures in that vertical dimension, in order for the plover chicks to take advantage of these very abundant synchronized emergences of tipilids in the breeding season for the birds. But of course, the ability of vertebrates to move uphill is limited by the availability of higher ground. And in fact, in the UK, golden plovers are now effectively restricted to the high tops of the highest mountains. Another kind of change in distribution is depth range shifts. We've already had a look at the behavior of earthworms as food for badgers. So in drier conditions, earthworms live deeper in the soil, and that has a demographic and population level impact on badger abundance and distribution. It's rather harder to pinpoint definite examples of equivalent vertical distribution changes, bathymetric changes in the marine environment, but there are certainly lots of examples of invertebrate shifts to deeper, cooler, and incidentally less acidic waters, but there aren't really any very clear examples of vertebrates as predators tracking those changes. And then finally, another very major class of response to climate impacting on vertebrates and mediated by invertebrates as prey are changes in phenology, the timing of biological events. And trophic mismatches between predators and their prey availability were first described in the marine environment. Here we have just one of several examples where temperature conditions, also oceanographic features such as wind speed and the resulting upwelling features, cause or influence the timing of phytoplankton blooms in the spring. The phytoplankton blooms are, of course, used as a trophic resource for zooplankton, and they, in turn, are foraged on by vertebrates such as fish. Now, the timing of those spring increases in air temperature and the timing of upwelling events has an impact on the timing of phytoplankton, and the zooplankton peak is basically tracking the phytoplankton peak. And ideally, of course, this then provides abundant foraging resources for fish larvae. But if the phytoplankton peak becomes much earlier in the year, then the zooplankton peak may be fading away before the fish larvae are hatching out. And this is the idea of a trophic mismatch, which appears to be driving population change both in the marine environment and also another well-described set of cascading impacts of climate in terrestrial ecosystems such as oak woodlands. Again, air temperature and rainfall have a major effect on the timing of leafing of oak trees, which of course are food for very hungry caterpillars, which we refer to in our lab as bird food, the birds here being pied flycatchers. Now, until recent decades, the oak leaf leafing event triggered a peak in caterpillar abundance in which in an oak ecosystem is a very concentrated peak of about 2 or 3 weeks duration. And so the timing of pied flycatcher breeding, egg laying and chick hatching needs to be very critically timed if those flycatchers are able to fledge their chicks effectively. But as the spring temperatures have warmed, oak leafing has become much earlier, so that the flycatchers arriving back from their migration from Africa, although it has become a little bit earlier, the flycatchers are now arriving way too late in many years to exploit this abundant peak of caterpillars as food availability. Again, it's another very well-described trophic mismatch. So what can we conclude from this kind of set of studies? Here's my local cultural reference, the Titanic being a Liverpudlian registered ship. We can, in various ways, looking around ecological examples, describe how the ship is sinking. And I'm sure you've all been to many talks that cast a kind of shadow of doom over future studies. And in understanding the impacts of these for us, in the words of Kate Winslet here, we can ask important ecological questions. Why is the ship sinking? Will the ship continue to sink? And crucially, can we stop the ship sinking? And mechanisms and understanding the role of invertebrates as mediating those population changes of vertebrates are very important. So invertebrates as bird food, other vertebrates are available, of course, are very important as mediators of climate impacts on vertebrate abundance, distribution, and phenology, as we've seen in this talk. And I'm willing to concede that invertebrates may be important in their own right. And in fact, of course, there's a whole book about this. And I'll leave you with the slightly disturbing sign here. Scott Johnson and Hevin Jones, thank you very much for editing this volume.
Thank you very much, Rob. Can we get that slide off?
Actually, I'll... oh, yeah.
We've got time for questions for Rob. So my question, you sort of touched on it there, about the temporal mismatches between invertebrates and vertebrates. Are there any examples that you can think of? Has anybody looked at this where they found that invertebrates that aren't quite susceptible to changes in the phenology could be used as an alternative food source for vertebrates? Or are we not even at that stage yet?
No, that's a very nice idea. And in fact, birds like flycatchers, they don't only breed in oak woodlands. And certainly the peak in caterpillar abundance in particular is so very pronounced in oak woodland because of the very rapid rate, I guess, at which oak leaves become filled with tannin and become much less good as food for the caterpillars. So in more mixed woodlands, there's a much broader peak of food availability, but actually a much lower peak as well. And Jess Smith in the audience is looking at those relative differences between woodlands in the timing, but also the total abundance of prey. And in other habitats as well, like wetlands, there's a much, much broader peak than you see in some of these classic mismatch habitats, as it were.
Okay. Any further questions? Okay, well, all this talk of trophic behaviour has made me quite hungry, actually. So we've got a coffee break now, but it looks like it might be mulled wine. I don't know, but we'll see. But anyway, I'd like to thank Rob, but all the speakers today. It's been a really fascinating and diverse thematic session. I've thoroughly enjoyed it, and I'm really grateful for the audience and also the speakers. Thanks very much.